Hi, welcome to my presentation. This is a presentation for the course Carbohydrates, and you are welcome to join me in this presentation where we talk about carbohydrates, what they are, how the plants use them, and what are the effects and what are the benefits and the applications in the industry. My name is Shrikant. I work for SLU. So the topic of today's presentation is the synthesis and mobilization of storage and structural carbohydrates. So this presentation is divided in 5 parts. First we will go what are carbohydrates, how they are synthesized in a photosynthetic cell, how they are transported and how they are allocated in reserve starch storage, how the starch is synthesized and degraded, what are the other plant carbohydrate storage products which are useful for humans, and what are the cell wall components in carbohydrates, how carbohydrates are used in synthesis of cell wall, and what is its composition. So let's get started. So what are carbohydrates? Carbohydrates could be divided into parts. Carbohydrates, that means hydrated carbons. So if we look at the structure of the carbohydrates, we see there are three elements in it: carbon, hydrogen, and oxygen. So the structure of carbohydrates is a repeating unit of carbon, hydrogen, and oxygen that is depicted as we can see this formula. Uh, so carbohydrates are mostly known as saccharides. It could be mono, di, oligo, or polysaccharides. What is important here that this repeating unit, I will just turn on my pointer. So this repeating unit, depending on how many units are there, it could be mono, di means two, oligo means more than two, and poly means too many of them. So it's basically a chain, and this unit is repeated in it. If you look at the explanation in Greek, I mean it's derived from a Greek word which is called saccharon, which means sugar. So all of the carbohydrates are usually sugars, and it has a suffix which is O. So if it is made up of glucose repeating units, then it will be glucose. So that means like wine or must. It derived from these words beginning from the Greek era. So we have different kind of carbohydrates or saccharides in plants. For example, fructose, which is predominantly in fruits. Sucrose, which you can find in sugarcane or beet. There is ribose, mylose, lactose. They are all examples of carbohydrates and predominantly saccharides. So we can see monosaccharides could be triose, tetrose, or pentose, or hexose, and so on. And what it means is how many carbon molecules are there in the molecule. So if it is 3, if it is 4, if it is 5, or if it is 6, and so on. So if you look at the structure of glucose, then you can see that usually in the solution material, it tries to make a ring. And that's why we see this hexagonal structure. It could be depicted as normal formula, where you can see there are almost 6 carbons, because glucose is a hexose, and it comes and forms a ring here. If we look at the Wikipedia description, so it just summarizes what we already talked about. Carbohydrates are sugars. They could be oligo or poly. It depends how many repeating units are there in it. There could be different type of sugars, which the most prominent ones are listed in this table. They could be glucose repeating units, galactose, fructose, xylose as monosaccharides if there is only one molecule of the repeating unit. It could be two, like for example sucrose, which has glucose and fructose. Let me just turn on my pointer again. So disaccharides, for example, sucrose is a repeating unit of glucose and fructose. And there could be polyols, means there are more than two repeating units, for example, sorbitol or mannitol. And then what kind of repeating units are there in it? So malto-oligosaccharides is maltodextrins, for example, that's a component made up of that. And starch, which is the main important molecule here, which we are talking about, could be of two types. It could be amylose or amylospectin, and it could be modified in a way to meet our requirements in industry and agriculture as well. You might have heard about non-starch polysaccharides, that's like for example cellulose, hemiglucose and others. We will come back to that later on in the slide. So moving on, carbohydrates are synthesized in plants. So the synthesis of carbohydrate is a spatiotemporal process, which means that it is highly compartmentalized into organelles. They are specific plant cell organelles which synthesize carbohydrates. And for example, chloroplast, if we have a look at the potato plant, you can see that the chloroplasts are mostly in the leaves. If we zoom in, then if you look at the structure of the leaf cells, all these green molecules, green dots, are chloroplast, and they are specialized in synthesizing of carbohydrates and starch. And there is a relationship between source and sink. What does it mean is like the carbohydrates are synthesized in the plant leaf cells, and they are transported through the vascular tissue to the storage or sink organs, for example, tubers in potato, where they are allocated or stored into the amyloblast cells of the tubers. So what these pink molecules you see, they are starch granules, and the blue outside cells are amyloblast. And temporal, what does it mean by temporal? It means it is time dependent. That means the synthesis happens during the daylight time when there is light and leaves could synthesize carbohydrates. During the night time, it is broken down into sucrose, transported through the vascular tissue. So in the morning and during the daytime, the chloroplast synthesize starch, then it breaks down and transport through the vascular tissue, and then again synthesized into starch granules, and the granules grow up from in the myeloblast. So it is a diurnal cycle. That means it repeats every day. As long as there is light, the carbohydrates will be synthesized. If we look at in more detail what happens in diurnal cycle. So this is a cartoon representing uh, a plant cell in the leaves. So you can see it has a chloroplast, and this is cytoplasm, and this is vacuole. So during photosynthesis, the triose phosphates are produced by the Calvin cycle, and it is converted into starch. Or these triose phosphates could also be transported to the cytoplasm, where they are converted into hexose phosphates, and it could be converted into sucrose. Now, as we remember, sucrose is glucose and fructose together. The sucrose is transported to the upper blast of the cells, and then, or coming back to the vacuum, or the sucrose could be transported into the vacuum and stored as a sucrose or fructose. Sucrose again is glucose and fructose. Otherwise, during the nighttime, this starch in the vacuum is broken down, and even in the chloroplast, it is broken down into glucose and maltose. And there are specific transporters which transport glucose and maltose out of chloroplast and convert into hexose phosphate, which is again converted into sucrose, and that is transported to the apoplastic region of the cell again. This apoplastic region transport of sucrose leads to transport of sucrose through the vascular bundles to the zinc tissue. And as we discussed, the zinc tissue is tubular. So going into more details about it, if you look at during the daytime, triphosphate could be converted into starch from the Calvin cycle. And this is just an example of Calvin cycle. If I just quickly go through it, you don't need to remember all of this, you can read it through the books anyways. So Calvin cycle leads to starch synthesis, and this is transported by triphosphate as triphosphate by the pyrophosphate translocator, which takes a pyrophosphate in turn to the chloroplast when it is moving the triphosphate out to the cytosol, which is converted into fructose 1,6-biphosphate, and by the steps of various enzymes is converted into glucose phosphate. This glucose phosphate is then converted into UDP glucose, which is the charged glucose molecule, and fructose 6-phosphate and UDP glucose is converted into sucrose 6-phosphate by sucrose phosphate synthase. This is then dephosphated. Sucrose phosphate phosphatase is the pyrophosphate is removed, and the sucrose is stable now. Again, all of these pyrophosphates accumulate in cytosol, and then they are transported into the chloroplast by the action of pyrophosphate translocator. So it exports triphosphate into the cytosol, and by then it takes the pyrophosphate back into the chloroplast. In this way, it maintains the photosynthesis in the Calvin cycle. As we know that sucrose is synthesized in cytosol by the actions of various enzymes, but there is a homeostasis between the concentration of fructose and sucrose. If I just quickly skip the slide to get an overview. So during the daytime, the triphosphate, which is 3-phosphoglyceraldehyde or dihydroxyacetone phosphate, transported into triphosphate to the cytosol. However, the concentration of these molecules are interdependent, and they're controlling the way or the direction of the reaction. So it could be either gluconeogenesis, that means synthesis of glucose and sucrose, or glucose degradation, that is glycolysis. All these reactions are feedback loops, and they try to maintain the concentration of various molecules in order to plant to survive during the daytime. This dihydroxyacetone phosphate is very important in maintaining the photosynthesis homeostasis. It can also be converted into glyceraldehyde 3-phosphate, which leads to the synthesis of lipids, which we might come later on in the presentation. There we're just explaining the same information which we talked about before. So here glucose 1-phosphate is converted into UDP glucose, and it leads to generation of 4 pyrophosphates, which are transported back to the chloroplast. Whereas if we say that sucrose 6-phosphate converted into sucrose by sucrose phosphatase. So in a nutshell, this is an overview of the photosynthetic process happening during the daytime. The carbohydrates are incorporated into triose sugars, which could be converted into, so just my pointer is on now. So carbon dioxide, carbon molecules are imported into the plant. They could be converted into triose phosphate sugars or hexose phosphate sugar, starch and glucose and fructose. Or during the daytime, this reaction of Calvin cycle, but it's dependent on pyrophosphate. So if there is too much pyrophosphate in the cytosol, it affects the photosynthesis process, but it is maintained by the plant in order to synthesize sucrose to store the energy or break it down to use more photosynthesis. That was the first session. Now we know how the carbohydrates are synthesized, and the next slides we will look at how these synthesized carbohydrates are transported to the sink tissue and stored there. So that's called carbon allocation.